But we have a cell growing exponentially, starting from some size Lk, and then dividing to produce a progeny of size Lk plus 1, adding some volume delta. And you can pick one of the progeny and start this process over again, forming what is called a cell size lineage history. And the thing that I want to draw your attention to is that you'll notice that these birth lengths are pretty even, right? It's fairly stable. So the machinery isn't perfect. For whatever reason, things can get bumped around, and it rather quickly can correct this. And these are called cell size homeostatic mechanisms. There are two main mechanisms that people talk about in the literature. One is called sizer, the other one's called adder. And the sizer mechanism is characterized by, regardless of what size you're born at, you always have some target size that you're dividing at. Okay? You always have some like pre-programmed size that you're dividing at. The adder mechanism, if you're born, for example, uncharacteristically short, you're still going to add the same volume before division. Right? So these are differentiated. So the thing that was kind of surprising is if you look, like this is a very hard thing to actually examine, right? We're talking about like sub-micron kind of behavior. But when you looked across the different domains of life, adder was very prevalent. So as examples, I have bacteria, human cancer cells, and then archaea. And you see that, for example, with bacterium, regardless of your birth length, you're adding the same length before division. Okay, so that's the main question that I'm trying to focus on, is what advantage might this have in nature, right? We're observing it so often. This kind of looks like it might be convergent evolution. Like, why would we be observing this, right? So we're not the first people to try to ask this question. This has been a question for a while. A lot of times when people try to measure the fitness effect for both these mechanisms, they don't see something that's like super strong. You'll see some effect, but maybe not something that's very big. And so we're wanting to take these mechanisms. We want to try to tie them directly to some metric for fitness. In this case, it'll be the asymptotic growth rate of the population. And then because this is something that we want to be very generic, whatever assumptions we make, we don't want them to be specific to like E. coli, for example, or any certain machinery. Right? So this is really meant to attack imagining if you, like the kind of a proto-cell before you actually had what's evolved today. So we don't want to make too many assumptions about the actual machinery itself. So broadly, what essentially we're trying to do is we're trying to relate this lineage level phenomena. So this is a whole bunch of cell size lineage histories overlapping to population statistics. So like birth length distribution, asymptotic growth rate of the population. So I do make one assumption very early on. It makes the math considerably nicer. So I assume that division is perfectly even. And I'm assuming that the elongation rate, or the rate that they're accumulating volume, is fixed. Okay. So the reason why that's so nice to assume is that it basically makes this kind of peak behavior go away. So I can turn these kind of annoying, exponential kind of peaks that drop. I can turn that into me just being concerned with 2 points, the birth length at the start and the birth length in the next generation, the start of the next generation. So this is an assumption that seems pretty strong, but in general, you can actually relax this and you'll get something that's qualitatively very similar. It doesn't change the result massively. But it means that I can characterize every step with a single conditional probability that allows me to just go from one birth size to the next. So the kind of natural thing to do, set up an integral equation like this. And here, my kernel is just going to tell me the expected number of cells in this transition. And this isn't quite ideal yet, right? So on the right, I have this discounted count, right? Because I have to account for the fact that this is further back in time. And then I'm relating it to the steady state, like birth length in some future point or the next generation. And the thing that you would want is to be able to just read off the top eigenvalue, and that would be associated with the growth. But because I have this dependence of tau on the birth lengths, I don't actually get immediately that I can just look at the leading eigenvalue. But with that symmetry assumption, that stops being a problem. Okay, so you can imagine, you could either view this as me computing like the spectral radius, or you can think of it like I'm building a lineage, right? Just applying, going one generation to the next, to the next, to the next. Integrate both sides, use this symmetry assumption, boundary terms die, and I basically just end up with this kind of nicer thing that I'm looking for. And alpha here is literally just the log of the like top eigenvalue. So it's just essentially I can now just focus on the eigenvalue. And it's directly related to fitness, you can see on that. There's an immediate relationship between the two. Okay, so just to quickly summarize, I basically am making this assumption And then I very quickly get this nice relationship for fitness, and I want to emphasize it's directly related to like the asymptotic growth rate. Weird going on there. Okay, so I haven't really talked about how I want to incorporate these mechanisms or this machinery into this model. So one way to measure them, I'm here to talk about a different one than this, but one way to kind of measure which mechanism you have within a population is you look at the birth size distribution, the added size, you do an align to best fit, and it's very simple kind of transformation. You can relate that to the correlations on birth lengths, right? And I'm interested in that because my whole theory is characterized in terms of birth lengths. So when you do it this way, Sizer has birth length correlations of 0, and the adder mechanism has birth length correlations of half. Okay, so I think the most kind of obvious thing to consider for these transitions is just a conditional bivariate Gaussian. So those correlations are what I was just referring to. Then I'm going to incorporate this lineage mean size. So you can think of it like the characteristic size that you'd measure a lineage at, or a lineage is kind of programmed to produce progeny at, in a sense. And then the standard deviation, so how error-prone this mechanism is constructing a lineage. And then when I do all that, this will be my kernel, right? So I have these transitions, and then I get 2 cells per division. So that's all I end up with. So if I plug this into the framework, it ends up really not, it gives me kind of a nice thing to see, I guess, as a check. But when I plug it in, effectively, it shows you that it doesn't really matter what the machinery is that's constructing these lineages, right? So if you look on the far right, I have this product that just gives me like a weight of the probability of different paths, right? And essentially all that I'm going to get when I look at this is just going to be log of 2, right? So I'll get alpha is log of 2. I can plug that in, and I get this result that Ariel was talking about before, where I just have the asymptotic growth rate equals the population like elongation rate or volume accumulation rate. This naturally makes you wonder, okay, under this framework, you're really just emphasizing there isn't a big difference between these mechanisms. So like, why would the size control method matter at all? And I think this is when we included mortality. I think it's a very natural thing to include in this type of problem. So the kind of impetus for thinking about adding mortality was, if you think about it, there really isn't, like, why do you, I guess, care so much about asymptotic growth rate or like population count, right? And in my view, the reason why that matters more than just like, I guess, accumulating mass or just having a blob that takes over a dish, right, which would be the alternative. The reason why I think you care about the population count is because it decouples death probability, right? So if I had a single blob that grew really fast and took over a whole dish, if that dies, the whole thing's done. But if I have a bunch of cells, I'm kind of bet hedging, right? That was the thinking that went behind doing this, is that the whole reason why you seem to want to divide at all is this kind of something like this. And so we came up with like a very simple, super minimalistic model that's basically keeping you away from 2 extremes. So here I have a Heaviside function. It's giving me this lower boundary. And I'm saying that you're not going to get a cell that survives below a certain size. So I have some minimal size. And you can just imagine you need like a certain volume for your DNA and ribosomes and stuff like that. Going the other direction, I'm essentially assuming that you have this constant, like every increment of volume that you add, you have this potential risk of death. So you could imagine you might make a mistake building your membrane or something like that. And how that gets incorporated is pretty straightforward. Just throwing it into my kernel. And when I do this, I get something like, right, you can see that I'm trying to invoke like this large deviation theory kind of stuff. Unfortunately, it's not just a straightforward application of like this lower boundary screws it all up. So you can't just quickly read off an answer. However, if you are in the case without correlations, you absolutely can, right? So the Sizer case is the one without correlations. We can right away get a nice answer, right? Instead of an integral of a product, it just becomes a product of these integrals. And this becomes very easy to solve. So focusing on the Sizer case first, I just want to point out kind of how this relates to this environment that we set up. So here I'm changing this characteristic size. So you're imagining this is like the kind of target birth size for the population or for these lineages. And going right to left, basically if you're on this extreme, you're suffering this like beta cost, right? And then as you go towards the boundary, you reach some optimum, and then near the boundary, those deviations or those mistakes in your division mean that you can just die. The populations will collapse. So then the question is like, well, how does the adder mechanism handle this, right? We're really interested in this comparison between adder and sizer. And what's a pretty nice, kind of surprising result, I think, is that the adder mechanism does quite well on that boundary, right? So we see that somehow, somewhat miraculously, this adder mechanism is actually able to survive below the boundary. So what this means, to be clear, is every single cell in the lineage, it will die if it's born below the boundary. But the characteristic size, the target size, is what can be below, and the population still lives. So this is a numerical solution. It would be nice to actually have a better understanding of what's happening. Okay, so our objective with this kind of minimal model was to obtain a solution that's very interpretable and could be understood. So I talked about the kind of large deviation theory part being hard. You can soften the boundary and work on it that way, and you can get a solution that's rather nice, but that's not what I'm going to actually talk about, because it's not really the type of thing we're interested in. We want something that's a nice, tractable, and understandable solution. Yes? Yes. You're talking about when, yeah. You'll see. You'll see. I'll show you. I will answer that question. That's exactly the question that it leads to. But yeah, so we get this, fairly nice effect. And again, we want to understand the effect. Is there other simple explanation, right? So you imagine if you have your target size below this boundary or at this boundary, if you have a sizer lineage, these are really not correlated, right? So it's every generation just trying to divide at exactly this target size. So that's just killing the cells in droves. These lineages just collapse. With the adder lineages, because you have these correlations, they're able to kind of maintain these runs. Okay. So if you have some cell that's just incidentally born above this boundary, they can have this massive fitness advantage. So the kind of simple approximation scheme that we came up with, again, this is the kind of trying to come up with something very nice and just immediately tractable, was the main difference between these is the sizer mechanism, again, is very flexible, almost you could imagine flexible rope or something. These are very uncorrelated. The adder mechanism has this kind of persistence. The kind of simple idea was, hey, what if we just add almost plastic beads on a rope? What if we just add these to kind of encourage it to have that kind of rigidity or these correlations? And that is quite easy to solve, right? We're just switching between something that's completely not correlated to these perfectly correlated blocks. So I'm effectively have an uncorrelated process on blocks. I imagine this is an approximation that's used in other things, but I don't know. I wasn't familiar with it. And then the only thing that's left is we have one kind of parameter, seemingly that's just kind of free, which is the block length, R. And the first thing that I really just thought to try is very simple, was what if we just talk about just averages? So it's just what would be the average correlations for some, for this kind of Markov process where each step has correlations row. And then what if we compared it to the correlations you'd have if you had some n-size population and R of them are perfectly correlated. And when you match them, you get this. There are other ways to derive different values for R, different arguments that you could make. But I think this one's rather simple. And when I do this, you get something that is a surprisingly accurate fit. So I wasn't sure how good this would be. It seemed like it was a very quick kind of attempt. So I guess starting with the right, you see that I think something that's kind of an interesting effect is if you're above that boundary, having an increase in these kind of errors in your ability to control your size, that actually causes the depression in your growth rate. But as you go below the boundary, those actually become advantageous, right? Which I think is kind of cool. It's also, I think, rather intuitive, right? If you have a target maladaptive size, mistakes are no longer bad, right? They're now good. You have a target maladaptive size. And then on the left is kind of it's a bigger kind of thing. This is what Ariel was talking about, right? It's basically, I have rho equals 0 is sizer, oops. But you can tune this, right? So you can change rho to be other things. Adder would be 0.5, but what happens if you go higher than that, right? It seems like you just do better. And indeed, that's true. So the kind of simple argument that I give, it seems like just the higher your correlations, the better you do. So I guess I want to point out that even if we stopped right here, I think this is a pretty nice result, because it's basically showing It's a pretty nice way to say why we would observe, like why adder would be more prevalent than sizer. Basically, it would no longer be kind of an even playing field. And it changes the question to be like, why isn't it evolving something that's even stronger than the adder mechanism? So in order to address what would prevent you from going towards a super strong or high correlation mechanism, we actually do look at essentially more of this kind of mutational argument. So here, this contour plot is showing me that approximation that I was showing you before. And so it's going to give me the growth rate of the population. This axis, z, is the number of standard deviations you are away from the boundary. And then this is that r parameter, which is kind of like a form of memory for the population. And this is the kind of closest we observed anything going towards the boundary. Okay? So the contour sizes are actually the fitness effect size of a mutation. So in order to look at like the way you kind of read this is if you were at this size, right, and you had a mutation to increase your R, you would need to cross at least one contour line to expect a net gain, right? If you don't cross at least one contour line, that mutation cost you more than you've gained in some sort of growth improvement. You really see that it doesn't actually matter until you get closer to this regime. But then it seems like you actually get these, you have these diminishing returns in the improvement in your growth rate as you increase these correlations. And then you reach a point where it's kind of saturated. So increasing these correlations, it does help you technically, but it's not as beneficial as that mutation will cost you. And so it ends up easing up to something that's slightly above an adder mechanism. So adder's 0.5, I think this would be 0.66. And a lot of times we actually do measure things that are slightly above as well. So this is kind of a loose explanation for why you would anticipate it not evolving to like perfection. Okay. So the last thing I really want to show you is, even though this was kind of starting off as like a toy minimal model, we were able to make comparisons with actual experimental data, which I think was quite nice. So every single data point here is a different experiment. These are different conditions for this bacterium. And then what we're looking at is the kind of empirical birth mean and the empirical standard deviation measured from those populations. And then the gray is the line of best fit. And then the black dashed line is actually what you can solve for, what would be the optimum, and using that, that approximation I was talking about before. And so the idea is that you could fit, and then you could try to find L-min and beta, right? And I really wasn't expecting too much out of that. Like I said, it was kind of starting off as a toy model. But we ended up getting like a pretty excellent agreement with experimental data. So this is what this approximation or this like fit suggests the kind of minimal cell size should be. And I don't know if you guys have read, there's like this paper by ESI June that essentially talks about cells. It's like a generalization of the MSK growth law, but it's like cells having like this fundamental unit of size. And he measures this as being the fundamental unit of size. So it'd be related to what you'd expect some sort of minimal to be. This also is very close to what we would measure, like what we see when we actually look at experimental data and we're looking at like lower thresholds, where we see the population kind of die off. And then there aren't a lot of data sets that we could find that actually would give us some viable thing that we could use to relate to beta. But this fit did relate to the limited data that we could find for E. coli. Again, just in a loose way, it fit as in like it was the same, like an order of magnitude estimate, right? So okay, so I guess just as a quick recap, essentially the question was why would adder have this advantage in nature? And then the argument is that these correlations kind of help the lineages survive. And then going the other direction, this benefit has diminishing returns. So the cost of a mutation would eclipse the gain that you would get for improving these correlations at some point. And then what I'm showing here at the end is this is just the numerics related to and compared to simulations. And then this is also, I think what's kind of nice to see is that looking at simulations, you get to see there's this kind of shift that adder has in terms of its birth length distribution, which I think is kind of fun. So yeah, any questions?